Hi everyone and welcome back to Advanced Higher Biology. Today we're going to finish off Unit 3, Investigative Biology, by looking at the third and final key area, reporting and critical evaluation of biological research. So, although that's quite a wordy title, as I've mentioned before, Unit 3 is seen as a bit of a half unit. It's more about the theory behind biological research, behind how biological research is carried out, how it's designed, and how it is shared with an audience as well. So again, this is going to be going through, as the title suggests, how you actually report back the information from this experiment that you have planned and designed. This is handy in terms of how all biological research is used, but will also be really handy for you planning out and writing up your projects, for example, as well. So we're going to start off just going through some basic information in terms of what you actually expect from any form of biological research and how it is written up. So in any form of scientific report, if you go and look at some online papers, go and Google Scholar, look at some scientific journals, particularly when you're researching your project, you'll find that scientific reports will have essentially a front cover. Page 1 will have a title. That title should explain exactly what the research is about. It should have something called an abstract, which is usually a paragraph or two right at the start that summarizes everything, essentially. It tells you the aim and a brief overview of the findings, and then there'll be an introduction as well. So as I said, you can see this on any form of published scientific reports, but it's also something you'll be carrying out in your project as well. So just so you're aware of all these different parts, you have, first of all, your title up the top, abstract in the middle, and down the bottom here you can see the beginnings of an introduction. So, as I said, your title has to give a succinct explanation of the study. Now, there's some examples on the right here, but at the end of the day, every title needs to tell you exactly what is going on. You can't have a title that is too vague, but you also don't want anything that is too wordy as well. You should be able to look at the title of any report and be able to tell exactly what this is all about. Nice and straightforward. Now again, an abstract, you may have came across this before, but it is essentially a summary of what your study is about. So for example, if you look at a scientific journal, if you look at a paper and there's a title that's specific to something you're researching, by scanning the abstract, you should really get a lot of the information that you need. What was the aim of the study? What did they actually set out to investigate? And what was the conclusion? What was the findings of this? And then after that, you can go into the paper and look at how they design this in more detail about their research and their results, but the abstract gives you everything you need. And again, that's something that will be expected from your project. Your introduction as well, though, is going to really set the ground for the background of all this research, and including why is this important. There's going to be a use of sources, which is something we're going to talk about later on as well, and supporting statements and citations. So this is more to do with referencing. So talking about background information, similar studies, similar results from other scientists, and referencing their work. So giving them credit for their work and citing them throughout the text. So the other thing you'll come across in the introduction is there will be some information about why this was carried out. What's the overall discussion about this? And again, when you're looking at your projects, you'll have big background information on whatever it is you're studying, but also the biological significance, why this is actually important. And that's the sort of this part here at the end that says placing the study in the context of existing understanding. What do we know already about this subject? How is your study going to help or assist our knowledge? How is it going to develop the knowledge of this? Like I said, one of the things that will come in here, and this will hopefully be something you'll do in class, I might do a separate video on this part as well, is looking at sources and looking at citations. So there's different ways of doing this. There's also websites you can use online, but essentially any information you've gained, either from a paper, from a book, from a website, these all have to be referenced at the end of your study and then cited throughout the text, sort of in a more abbreviated form, like the author and the year. And that needs to be in a standard form, so people can look at this and look at where you got that information from and make sure it's all correct. So examples of this are these ones here. Your reference list should be in alphabetical order. And again, your references are going to be at the end. So what you'll tend to find is this is an example called Harvard referencing, which is a really common way of referencing any information you find. So for example, there's a bunch of titles here. So you'll have your authors first in terms of their surname and then their initials. You'll have the year of publication. You'll then have the title of that paper as well. That's normally finished by or followed by the name of the journal. Here it's in italics, the volume of that journal in bold, and the page numbers. So for example, you can have a look at one of these and find where all these papers are, and it's a way of crediting where you got that information from, but then also citing it through the text so you can go and look back in it. Again, there's other ways of doing this. It doesn't need to be exactly this. This is probably the most common way of doing it. And again, we might do a bit more information, an example later on. If you want to give this a go though, this is something that I've just made up to try and help you with Harvard referencing. So again, the surname of the author or authors. In this case here, in brackets, you have the year, the title of the article, the journal in italics, the volume in bold, with colon at the end, and then the page or page range as well. Again, we might do this in a different video as well, but you should have references at the end and citations throughout your text. Now, in terms of how you report and evaluate your experimental design, you'll have a method section. Now, in National 5 and higher, you may remember you used to just do a brief method. It was a very basic summary of how you conducted your experiment, and you didn't go into any detail at all. So there's a few different things here. First of all, your experimental design has to address what the aim of your experiment is. There needs to be a thought process behind, could this experiment actually test this hypothesis, and does it test the aim? Is it linked in any way at all? The other way you'll need to look at your experiment as well is you need to essentially defend all your decisions, and really you'll be defending the validity and the reliability of your experiment. So again, if you're not actually testing your hypothesis, if your experimental design is not relevant, or if it's a bit vague and you're just making a jump saying this result means that X, then that's going to be an invalid experiment. But you also need to be looking at your reliability. So as always, have a look at your sample sizes, but also validity as well. Is this going to actually work? Have you planned this out? One thing we've mentioned in this Unit 3 as well is the idea of your controls and your confounding variables. So again, in your project and in any other research, you really need to take account of what is the control. Is it a valid control for your experiment to prove that any change in the dependent variable is caused by the independent variable? You also need to be looking at any form of confounding variables. Now, confounding variables, you've been doing questions like this since National 5, essentially, looking at an experiment saying what needs to be the same between all these different test tubes, for example. So those are all the same cases, but also particularly if you're working with people or animals, there's a lot of other variables that can have an impact on your study. You need to address them, you need to standardize them as well if there's a way of not fully controlling them. So again, defend your decisions every single time. So as we said, if a confounding variable is going to have an impact, that then calls into question the entire validity of your experiment. So have a look at what's going on. Don't just look at how you've set up your experiment, what else is going on within that experiment, within the room, the time of day that could be having an impact on that. You have to address these and defend these at every case. In terms of bias, we had a little look at sampling the other day as well. So selection bias is if you select your sample in a non-random way. So remember going back to your whole idea of a representative sample. If you're sampling in a non-random way, if you're choosing, I want to pick that plant and that plant and that plant. Think of it like throwing quadrats into the bunch of daisies that you know are going to skew your results. That is not going to give you a representative sample of the whole population, and again that's going to have a real impact on your study. So maybe look at how you can randomize if you are having to select certain things as well. Sample size again is something we've discussed all the way back from National 5. If you decide you're doing a study on, say, the effect of caffeine on the heart rate of Daphnia, and you do this on 3 Daphnia, and you say, oh right, this has had an impact, that's not sufficient. You need to have a larger sample size. There's always going to be bias if you just have a couple of Daphnia. If you have 5 plants, you need to decide what is a suitable sample size, and again, the larger is going to be better for you. In terms of your data analysis, whenever you're carrying out your studies, you're going to have a table of results, and then there will be some form of appropriate use of graphs, or maybe box plots, charts, whatever it is you're using in terms of showing your results and interpreting the data from them as well. Now, as always, the key thing here is it's not about making things look complicated and look like you've put a huge amount of work into it all. Data should always be presented in a clear, logical manner for analysis. You should be able to have a look at results and very clearly say what was the result. Was there an impact? Was there not an impact? There might be huge outliers. There might be results that are a bit mad. That's down to you later on to defend or explain that in your analysis, but your results should be nice, clear, and normally some good form of graphical representation as well. Statistical tests, you're not required to carry out any form of statistical test in your project, but it's good to know what they're there for. So essentially, whenever you're carrying out an experiment, there's always that chance that the differences between your two samples have entirely occurred by chance. Now, if you have a large sample size, if you have large or differences in standard deviation, then that is not really going to be the case. But you can use statistics to basically run the numbers and see what are the chances that these differences occurred by chance. So again, it's something you'll probably come across more if you go into biology in university, but statistical tests are there to prove that this change in your dependent variable did not occur by chance. If you have what we call a statistically significant result, that means that it is very, very unlikely that whatever difference you have found was due to chance. And again, you'll be able to see this often in your graphs as well. A way of showing that variability that we've discussed as well is error bars. So again, this has been mentioned beforehand, but you can see 2 different examples of error bars here. In the graph on the bottom left, you can see the error bars, and that's showing the variability of data around the mean of which the bar graph is constructed from. And you can see there's quite clear differences between the different plant species there on the left-hand side. There's pretty big differences on the orange bar graph on the bottom right as well, but you can see particularly that Solanum tripotatum on the right-hand side has got a massive error bar. So in that case, the differences that you've seen that look significant may not be as significant as they seem, because you have a huge variability going around there. So again, whenever you're looking for significant differences, you do not want error bars to overlap. Once you have your results and you've explained your method and you've shown your background knowledge in your introduction, you're going to then evaluate your results and conclude your results, always referring back to your aim. So remember, whatever your aim is, that is what your experimental design was set out to test, that's what your results should be showing, and that's what your conclusion should be talking about. Always be careful you refer back to the aim. Even in Advanced Higher, there's always questions that say what was the aim of this experiment, or give a suitable conclusion for this experiment. If there's a study that's used in experimental design that has not directly tested the aim, your conclusion still has to refer back to the aim. You can't just say the number of bubbles increased, or an increase in light intensity increased the number of oxygen bubbles. If the aim was about the rate of photosynthesis, you must refer to the rate of photosynthesis, for example. In your evaluation, you're going to defend essentially and explain your validity and reliability of your design. So why did you use this protocol? How was the procedure relevant? How did it do the best that you could in this experiment? You should also look as well at this idea of correlation and causation, which we've mentioned before as well. You cannot just simply say the graph went up, therefore this X definitely affected Y. People can be very tempted to say that because your results look good and that must be the reason. That's the correlation. You can state the correlation, but that does not mean that is the cause. And you can maybe explain if there's something else that may have caused that also. It's always good whenever you have end results as well to tie your results into existing investigations or your existing knowledge. So often you'll have an experiment that will fit the background biology. You'll say, well, this result makes sense. This is what should have happened because, temperature was at optimum, something like this. But also look at other people's studies as well. You may have found that other people have found the same thing as you. You may have found that people have found opposite results. Both of those are equally valid. You can explain why. You can maybe look at differences or similarities in their studies and compare it to your result as well. Finally, when you look at the evaluation then, the whole point of any scientific sharing or investigation is really to show an awareness of your contribution of scientific research. Now, the point of everything is to increase scientific knowledge, and this is quite a nice diagram here showing the impact of every study that is done. Now, this one's maybe not entirely relevant because it's talking from the change from school to postgraduate level, but even at school level, even at advanced higher, what you're doing in your project should really be something new. It should be developing some new form of understanding based on an observation you've had. And the idea of science is everyone is sharing their investigations or sharing their research and their findings to try and increase our scientific knowledge. And that should have a beneficial impact to the social, economic and industrial life of our community as well. So you can see in this diagram we have the current knowledge boundary. Every study that you do is going to push on that boundary just a little bit more and eventually break through what the current knowledge is. And the idea of all these individual investigations and going back to key area 1, the reason why all this information is shared is so that we can all actually work together in order to improve our understanding of scientific processes. So that is all for Unit 3. As I've said, it's quite a quick unit. It is a bit dry, it's not the most exciting, but in particular, when you're going and writing up your project, which probably quite a lot of you are doing at the moment, it's maybe worth going through all these details again, just to kind of check, have you actually shown the more basic scientific understanding throughout your project? So it's worth looking at. Also, there will be questions in your exam that will be based on your understanding of these, whether it's experimental design, whether it's the scientific method, types of sampling, that side of things as well. So hopefully you found these videos useful. I know quite a lot of you are asking for Unit 2 videos. The good news is Unit 2 videos are coming. As I explained in the end of the Unit 1 videos, I wanted to get Unit 3 done first, and then we'll go and finish off with Unit 2. It's not too long till the exam if you're listening to these as they come out. But we will get these done in time, and thanks so much for your patience. I do really appreciate all the messages you send as well. So thanks so much for listening, and good luck going through your research and if you're doing your project, and I'll see you for key area 1 of Unit 2 very soon. Bye for now.